All right. Well, thanks for the invitation. Thanks everybody for coming tonight. Brave of you, a professor. Oh my gosh, 7 o'clock at night. Like you really, I know there's entertainment alternatives. Let's just put it that way. So I gotta make this worth your time. So tonight I'm going to talk about some of the work that my lab is pursuing. If you're not a scientist, I hope to give you just a little glimpse into how we do what we do and discover things. And if you are a scientist, there'll be some red meat for you as well. Um, but first, and especially, I think this sort of topic, and if I think about the people I've met in the course of my life, this is often the area of snakes, herps, et cetera, is often an area of inspiration for a lot of biologists that I know, especially when they're kids. And so I want to take a moment to acknowledge why we do what we do, our personal motivations and inspirations. And one of my favorite writers is a French writer named Albert Camus, and I think he got it right when he said that a man's work is nothing but this slow trek to rediscover through the detours of art those two or three great and simple images in whose presence his heart first opened. And it's true of art. It's also definitely true of science. These are the images that opened my heart when I was a kid. Growing up in Toledo, Ohio, I started keeping snakes. I actually had a speckled king snake, the species on the left, but I had green snakes and garter snakes and a yellow rat snake, and I had salamanders and frogs and things like that. And it was these animals, and I think their beautiful spotted and striped patterns that inspired my interest in the animal kingdom and in pattern formation, and my love for the animal kingdom, which I've been able to enjoy both in and out of formal science. And I really do love the animal kingdom. I try to tell people that I love it all. I love the spiders, I love the snakes, I love, of course, the giraffes and the elephants and everything else. And my hope was to someday make a living studying or perhaps helping these creatures. But first, I had to make do with some smaller things to study, and I wound up becoming an indoor biologist. So that's going to disappoint some of you right now that I'm not a field herpetologist. I'm an indoor biologist. But I asked a big question, and that question was around this issue of novelty. It's one of the central questions in evolutionary biology, and it's occupied my lab for more than 20 years. And in fact, we've studied things like butterflies and how they make their spots. And these questions have to do with the kinds of genetic changes that underlie changes in morphology, or what we call sort of anatomy and form, and as well as new protein functions. Proteins are the things that do the work in the body, and we want to understand how new functions evolve. And if you think about it, first of all, this is a really important thing to the diversity of life. And if you think about creatures, maybe some of your favorites, they have distinguishing features, things that they have figured out how to do that other creatures have not. But this has also been a really important question in the whole arc of evolutionary biology. It's something that, in fact, troubled Darwin because it was just very hard, given the state of knowledge in the mid-19th century, to imagine, and he sympathized with his readers, it was really hard to imagine how new things came into existence in this gradual way that he described first in the origin of species. And particularly when you look at something as what they describe as sort of perfect form, like an eye or a wing, how did that come about gradually? What good was half a wing? What good was, part of an eye? But in modern biology, we have much stronger tools and a much bigger foundation for pursuing these questions. So I want to give you a little insight into how we study these things now. Novelty was defined by one of the major 20th century figures of evolution as a newly acquired structure or property which permits the performance of a new function, which in turn will open a new adaptive zone. So we're not just talking about changes in creatures, we're talking about things that are sort of game changers in creatures and that open up new lifestyles and new ways of living. And so when you see a creature like this, now those of you who have voluntarily tuned in tonight, I suspect that what most of you see is yes, a very dangerous creature. Some of you might avoid, some of you might be drawn toward, but I see an irresistible evolutionary mystery, because even just a quick glance at this animal reveals a host of novel anatomical features. May not think about it too much, but the limbless body, snakes evolved from lizards, from four-legged lizards, and their probably first as burrowing animals, and this limbless body is a much more efficient way to burrow. Of course, the rattle is famous to ward off danger, to tell other creatures, hey, I'm here, back off. The forked tongue, an amazing sense organ to help the rattlesnake and other snakes sense their environment. But rattlesnakes belong to a group of venomous snakes called pit vipers, because they have these heat sensing pits right in the front of their face, which are very sensitive to small changes in temperature and allows them, for example, to help hunt prey at night. But if you, when it opens its mouth, it's obvious there's even more to talk about. The fangs, which are modified teeth with a hollow center through which the venom is expelled. And of course, there's the venom itself. So I'm not going to talk about those anatomical features tonight. I'm really going to focus on venom. And if you just give it a moment's thought, you can think about all the times in the history of the animal kingdom that venom has been invented. Spiders invented venom, scorpions invented venom, jellyfish invented venom, octopi have invented venom, and of course, snakes have invented venom. And there's different types of venoms, and I'll get into that a little bit more as we go on tonight. But the venom, it's a great model for studying novelty. And one of the key challenges in being a scientist is you're trying to find examples of the general phenomenon that you want to understand. So there's all sorts of novelty in this world. You know, why is venom a particularly good way in? Well, the first is it's fairly recently evolved. Venomous snakes are fairly young as animal groups go. In fact, rattlesnakes, for example, are probably only about 20 million years old. And we understand that this is an evolved trait that enabled snakes to subdue their prey and avoid combat. So things like constrictors that remain in contact with their prey. Well, there's a danger in being in close contact with your prey. If you can ambush your prey, strike and pull back, and then the animal's dealing with this, the effects of the venom, that's a lower risk hunting strategy than physical combat. The venoms have been studied by biochemists for many, many decades. And so we understand there's a wide variety of toxin proteins in every venom, and there's different cocktails of toxins in different venoms of snakes across the world. And again, you'll hear about a couple of the more notorious ones in the course of tonight. And another reason for us to focus on this is we understand that snakes and their prey and their predators, and there are many things that feed on even pit vipers, they're in evolutionary arms races. And what we like about evolutionary arms races, and the analogy does come from the military, which is the prey, there's a lot of pressure for the prey to evolve resistance to venom. And as they do, that puts pressure back on the snakes to evolve new tricks in the venom. And that puts pressure back on the prey, et cetera. And you get this sort of feed forward mechanism. So you get sort of evolution on steroids. So we focused on rattlesnake venoms and on tracing the origin of certain toxins. And one very practical reason was there's been a fair amount of work on rattlesnakes, but here we are living in North America. It means we have easier access to material than we would to other sorts of snakes. And there's some practical reasons in that the pit vipers of North America, rattlesnakes, cottonmouths, water moccasins, they're of significant medical concern. So some of our work has some practical implications, which I'll get to in the last few minutes tonight. So let me introduce you a bit to venoms. And you may be a little familiar with rattlesnake venoms, but I'm going to just divide the venoms up into two major categories. And this is also generally true of venoms across the world. So many rattlesnake species venoms are hemorrhagic, and they disrupt hemostasis. Hemostasis just means it's sort of like the stability or equilibrium in the blood system. So they're messing up blood flow, blood pressure, blood clotting, et cetera. But the hemorrhagic part is the rattlesnake species actually cause hemorrhage. They cause blood vessels to open, and that's a big problem in terms of shock to the animal and for the spreading of toxins throughout the body. But there's also rattlesnakes that are neurotoxic, and they disrupt neurotransmission. And the danger there is, for example, respiratory arrest, that if the neurotoxins hit some of the central nervous system, that can start to shut the body down. And in fact, those neurotoxic rattlesnakes are drop for drop more lethal than the hemorrhagic ones. The hemorrhagic ones can cause, they can also kill people, but they can also cause a lot of tissue damage. So if you've known anyone that's been bitten by one of the hemorrhagic snakes, you know there's a lot of tissue damage. But the basic questions we want to understand is we, in general, want to understand something about these venoms, but of course, it means that not all rattlesnakes are the same, and there's been some inventing going on somewhere in the history of rattlesnakes. So the basic questions we want to study are, how did these venom toxins evolve? And the big question we're keeping in our mind, and you'll even get a decent answer to tonight, is, are they entirely new proteins? So essentially, did they evolve from scratch? Or did they evolve from non-toxic ancestral proteins, something that did something else? And the pursuit of these questions is very much like detective work. It may seem like a cliche or anything like that, but I still tell people to this day that science is very much detective work. You're playing hunches, you're looking for clues, you're kind of put these clues together, and you're testing them against what we call hypotheses. You're testing them against the ideas of how you think nature works. And sometimes you find things that are consistent with your idea of how something works, but you often find things that contradict it, and that helps you refine your idea. And that's how science moves forward. So these are sort of the two ideas we have in mind for the moment. And what are we going to do? And I promised you that I want to give you some insight into how do we do this detective work. And if you've not been exposed to this sort of approach, I'm going to give you a little bit of DNA forensics. So to understand how the venom toxins evolved, the clues are actually in the DNA. And so we want to look at how these toxins are encoded in DNA. And we want to ask, by comparing venomous snakes, in other words, those snakes that make these toxins, we compare their genomes, and genome is essentially the entire DNA library of the animal. That DNA library is sort of the recipe book for making the animal and all the things that the animal does. We compare that with those of venomous species and say, okay, what's the difference here between a venomous and a non-venomous snake, or even a venomous snake and all sorts of other relatives for animals with backbones, and say, what went on in snakes that gave them this power that other animals don't have? So this is, as I said, it's a forensic approach, and let me give you an idea of how we do it, show you what I mean. Okay, so these little ribbon diagrams that kind of look a little bit like, I don't know, I guess sort of the ribbons you take out at Christmas to wrap your packages in. But these are 3D models that scientists make of molecules that tell us how that molecule folds in three-dimensional space and how it interacts with other molecules and with tissues and things like this. And this is the model of a neurotoxin. This is a neurotoxin made by snakes like the Mojave rattlesnake. It's made by certain timber rattlers. Not all timber rattlers, but some timber rattlers make a neurotoxin like this. And certain other species in the American Southwest and in Central America and South America. So it's a really potent neurotoxin, and we want to know, where did it come from? Because not all rattlesnakes have it, and certainly most other snakes don't have it. So we went looking for it in the DNA of the Mojave rattlesnake, and this is the sort of thing that we make little schematics that show us the way things look in DNA. So what I'm showing you is a schematic of a stretch of a chromosome in the Mojave rattlesnake. And the diagram here with the various colored arrows facing different directions are the locations of genes. So genes are segments of DNA that encode a particular function. In this case, encode a protein. And the ones that I'm circling here are encoding the two pieces of the two proteins that when they come together, they form the neurotoxin. And the other things highlighted there in color are other very closely related genes, one of which there in light blue is another toxin made by these rattlesnakes that are unique to rattlesnakes. And the things that are in gray and white on the edge are genes that essentially all animals with backbones have. I'll give you a sense of this on the next slide. But I just want to give you a sense, you can sort of imagine the detective with the magnifying glass. Well, in our case, the magnifying glass is a computer. and we have the entire sequence of the DNA of this organism in the computer, and we're looking for where the toxins are encoded in the DNA and what's around them. Because when we look and then we compare with other animals, it's pretty clear what's going on in the history of rattlesnakes. So let me show you one of those comparisons. So you can think of this as sort of a tree, a tree of a bunch of relatives, increasingly distant relatives from the rattlesnake. So here along the bottom is a map of the genes of the Mojave rattlesnake, and I just showed you the two pieces of the neurotoxin here in orange and here in green. But if you compare this part of this section of this chromosome with all sorts of animals, other snakes, lizards, turtles, birds, mammals, etc., you can see so much is in common. All this stuff at the flanks in gray and white is stable through 400 million years of evolutionary time. And a lot of stuff in the middle is stable. You can see things like the yellow gene that's marked there in D that's shared among all these creatures. But what's really telltale, just jumps right out at us, is that in the rattlesnake, that in this area, this purple, where this purple G exists, this G gene exists in boas and king cobras, et cetera, this whole region's expanded and a whole bunch more genes are there. So this is the smoking gun. This is the hot spot of where invention has been taking place, and new information exists in this rattlesnake that doesn't exist in lots of other kinds of snakes and other kinds of animals. So we know, and in fact, it is the purple gene. This is the ancestral gene, and these toxins are derived from that ancestral gene. So we know that the toxin genes, they came from a non-toxin ancestral gene, or ancestral, we call these a locus. I should just refer to it as a gene. Okay? So I hope you got that. We're going to have another example, one or two more examples of that in the course of my talk. But I just want to get you a sense that, as I said, forensically, it's by seeing how things are encoded and comparing it with other animals that we can deduce what happened in the course of evolutionary time. And in fact, Matt Giorgiani in my lab group has traced this invention back. I just want to have a chance to tell you a little bit more about these pit vipers. So pit vipers, all the pit vipers, so that's water moccasins, copperheads, rattlesnakes here in North America, but it also involves things like bushmasters and fer-de-lance and all sorts of interesting snakes in Central and South America. All of those pit vipers have a common ancestor that came to the Americas about 24 million years ago. We can tell when we do an evolutionary tree of those animals, they all have one common trunk in their tree. And we know that their ancestors, there are pit vipers over in Asia, and that's a deeper branch of the tree. So what happened was, in the course of evolutionary time, is that some line of pit vipers made it over here to North America from Asia. And of course, you may know that's the story of lots of animal groups have gone one way or the other, Asia to the Americas, Americas to Asia, often through changes that are taking place geologically up here in this part of the world. But as diverse as all those snakes are that I just named in the Americas, they have a common ancestor that's only about 24 million years ago. And in evolutionary scale, that's pretty darn young. It's really been what we call a radiation. It's like these venomous snakes, their ancestor got to the Americas and just exploded in variety and diversity of species. And that's also really especially interesting to evolutionary biologists, because that means a lot of evolution has taken place in a relatively short period of time. And you can imagine that with these venomous animals coming in, and now they're feeding on all sorts of prey, their venom is evolving quite a great deal. So let me talk about the other kind of venom that we know about from pit vipers, and that's hemorrhagic venom. And I'm going to show you another one of those little ribbon diagrams. This is just something that scientists do. There's probably no way to sort of visualize in sort of a macro scale what these molecules are. But it's just to tell you that we know the anatomy of these molecules. But what we know about these metalloproteinases, and they're called metalloproteinases because they're proteinases, which means they digest proteins. So they're enzymes that break down proteins, and metallo means they use the metal ion zinc as part of that mechanism. You can see the zinc that's mapped right here. Now, what's particularly impressive about these molecules is that the hemorrhagic pattern is because they destroy the integrity of the vasculature. So when the snake bites and these toxins hit vessels, they can bust their way into and through vessels, and those vessels leak, and now you have hemorrhage going. And the hemorrhage can be quite profound. And if you've ever seen pictures of a human snake bite victim, like especially in the hands or the feet, it's a remarkable extent of hemorrhage. And also by breaking open the vasculature, you're also, the other toxins that are in the venom have sort of are gaining access to the circulation. So these are a really important class of toxins. So we thought, well, let's look at a rattlesnake that has, so it's almost kind of the classic form of a hemorrhagic rattlesnake, and that's the Western diamondback rattlesnake. Big range over south and western, especially the southwestern United States. Big animals, easy to get venom from these animals, but also a significant animal in terms of medical concern. So again, we looked at the chromosomes of the Western diamondback rattler. We dialed in. We said, okay, where are these metalloproteinases? And we were expecting to find, based on decades of work by biochemists, we thought we'd find about 8 or 9 metalloproteinase genes. We found almost 30. And this is just, smack in the face. It's saying that in this part of the DNA, this has gone crazy, that there's a whole bunch, a very large number of these venom metalloproteinase genes. And now when I do that same sort of comparison, and we look at other snakes and other animals, Matt's drawn this diagram here to just give you the idea that we can tell that there's an ancestral gene that is known as ADAM28. We even have it, so humans have it. And we find it in all these animals with backbones. But that gene was the forerunner. It was the ancestor to this massive expansion of metalloproteinase genes that became toxins in rattlesnakes, and a huge number in particular in the Western diamondback rattler. It turns out that no other snake we've looked at has as many metalloproteinase toxin genes as the Western diamondback. So you're now starting to get a picture of how we can trace the origin of these toxins. And I thought for fun, I would just tell you another little story of doing this tracing. And we're going to go all the way across the world. And I'm going to show you a little video from Australian television. And they're going to give a demonstration here of what one Australian snake does to the blood. And this is a third theme. I've talked about toxins that are neurotoxic and stop neural transmission. I've talked about toxins that are hemorrhagic. Well, here's a different story.
My blood is going to be envenomated.
So I'm going to show what cosotypan venom does to the blood. Now, of course, this is a smaller volume than what's in your body. So this will actually simulate what happens to prey items as opposed to inside a human body. So the first step is I'm going to pour one of these vials into one of these jars.
This is liquid gold here. Christina pours my blood into 2 jars. One will be mixed with venom and the other water as a control.
So that's going to prove that it's not just the fact that we've taken it out of your body.
And then it's toxin time.
So here is Squishy's venom that we extracted recently.
And in less than 3 minutes.
So as you can see, as I go to pour, so we can demonstrate what's actually going on.
Oh my God, it's really solid.
That one is turned into a piece of jelly, whereas the control is still liquid.
Here we go. Fascinating. That was so quick.
So this is another strategy for disrupting hemostasis, and it's not hemorrhage, it's not uncontrolled bleeding, it's uncontrolled clotting. So it's a procoagulant venom. Talk a little bit more about it. So just so you know, what the blood clotting system has been studied for many decades for lots of medical reasons, veterinary reasons, and others. So we know this coagulation system, which can get triggered both by external injury, so what we call the extrinsic pathway, or can actually get going on its own inside of a vessel. But there's really just, it's sort of a domino effect, where a bunch of proteins act in sequence to eventually create a clot. And I'm going to introduce the names of a couple of those. So it's factor 10 using the Roman numerals here. Both these pathways converge on factor 10. And another cofactor called factor 5 or 5A that works with it. And what they do is they act on yet another protein called prothrombin, an inactive form of the protein, converted to an active form called thrombin. And thrombin is what acts on another protein called fibrinogen, creates fibrin. And fibrin is actually, it's the fibrin, cross-linked fibrin that's key to the blood clot. So what I'm showing you here is this sort of domino system that creates the clot. And it's sort of an amplification mechanism. There's a lot of regulation in this system so that you don't have uncontrolled clotting. Yet it's also has to be a very rapid system because we've all had, you know, cuts, et cetera, and it's pretty marvelous how quickly we clot. So when there's an injury, you know, our blood clots. And you can imagine this system has been around for several 100 million years, and how valuable it is to animals with an internal circulation like us to clot. And so two keys in this system that sort of the regulated step that determines whether this is going forward or not at all are these proteins, factor 10, A for activated, and factor 5, also A for activated. And so these clotting factors have all these Roman numerals. I think factor 8 is the famous protein which is missing in people with hemophilia. So you can imagine if you're missing some of these components, you have blood coagulation disorders. One of the, I said the famous one being hemophilia. Now, okay, so these are really important proteins, but what was being demonstrated in that video is that there's a small group of Australian snakes that have really came up with a interesting trick. So these proteins are made in the liver, and they're in the circulation, and they're circulating at actually pretty low concentrations. And then they're going to be activated and cause this cascade of things that will create a clot. But one group of Aussie snakes, just think of this. This is, you know, it's either brilliant or sinister, depending upon your view of the world, have hijacked the clotting system as a new source of venom toxins. So these proteins, normally they restore hemostasis, right? If there's a break in a system, they clot, they plug the hole, right? Well, what these snakes have done, and this is a schematic of the venom gland and the fang, is they've hijacked factor 10 and factor 5. So that a version of those proteins are now made in the venom. And so they inject those proteins into prey, and they cause unregulated, and as you saw, very fast clotting in their prey. And this is an incredibly powerful, this is the very famous taipan and brown snakes of Australia. Drop for drop, it's 200 to 500 times more deadly than a rattlesnake venom. So this is an incredibly potent venom. And it's relatively recently evolved. This is just, Australia has lots and lots of snakes. They have lots and lots of venomous snakes, but it's just this group of taipans and browns that have come up with this dual action sort of trick and hijack these blood clotting factors. So how did they do it? Well, there were a lot of steps to it. And Yuri Van Thiel, a student in my lab, is working on the detail, but I'm just going to show you the two smoking guns. So what's happening here is that the normal proteins which restore hemostasis are now being used outside the body in prey to disrupt hemostasis. And we can see what's going on in these snakes. And these are the Latin names, but there we go. This is the taipan, this is the brown snake. And whereas other snakes have just one copy of the normal liver version of the factor 10 protein that works in normal blood coagulation, these snakes have come up with a second copy of the gene. One of those copies is dedicated to being expressed in the venom and take down the prey. So both the taipan has one extra copy. Actually, the brown snake has 2 copies of this gene. But anyway, same story you were essentially seeing in the rattlesnakes is that an ancestral protein has been co-opted, I'll define that term in a second, to a new function, in this case, as a venom protein. And this is what happened with factor 10, same story with factor 5. Again, these schematics are just showing us in the genomic map what's been duplicated. And whereas other snakes like the tiger snake or other snake species just have one copy that operates in the liver and does normal blood clotting, they have two copies of this gene, one of which is devoted to being made in the venom and injected into prey. Okay, so what all of these sort of forensic stories add up to is to tell you that the toxin gene complexes, the meaning of these, that the multiples of toxin genes that we often see, they're the product of co-option. And co-option is essentially, it means we're borrowing, we're using something that's operating in other tissues and other cells and expressing in the venom gland. So snake venom metalloproteinases, these PLA2s, which are the type of toxin I showed you for the neurotoxin, factor 10, factor 5, and essentially all venom toxin families. They're derived from ancestral physiological proteins, proteins used in the body, and they've been co-opted, recruited from other tissues to be expressed in the venom gland, and therefore, secreted outside the body into prey. And the other signature you've seen is that while they're derived from an ancestral gene, the duplications have taken place. More copies of that gene have built up, and those genes have been devoted to the venom function. And there's been further molecular innovation past that stage. So this is the way nature often invents, is that the ancestral functions maintained. Snakes have to have these proteins to clot. They have to have these enzymes to do other things. But the venom proteins are devoted to working in the venom gland and being injected into prey. So this is the big evolutionary idea, is co-option. And this is what we've been able to understand now from studying evolution at the DNA level over the last 20 years or so, is that nature largely invents, not at all from scratch, but by using existing things, existing traits, that might be a piece of anatomy, proteins, genetic elements, etc. So, and it kind of makes sense if you think about it. It's a lot easier to invent using existing things and new combinations of things than to generate them from scratch. Okay, so I'm going to just give you one more example because I think you might be curious about it. The evolutionary story is similar, but it's sort of like the flip side of the venom coin, and I hope you find it interesting. So one of the reasons we've been studying venomous snakes, as I said, they're prey, they're predators, they're in evolutionary arms races. So something like a rattlesnake can put a lot of pressure on the rodent prey. The prey evolve resistant, which in turn pressures the snake venom to evolve. So when we look just at different rattlesnakes, even rattlesnakes that live close to each other, their venoms can be pretty different. And that reflects they're working on different prey. Those prey are evolving resistance. It's a very dynamic situation. And I also told you that there are predators of these snakes, most famously, for example, things like king snakes that can eat pit vipers and evolve resistance to their venoms. So again, pressure on the predators in this case. But there's another third element. Maybe those of you who are snake fans, you may have heard about this, but it turns out that a lot of these snakes, rattlesnakes, certainly true, they're resistant to their own venom. And it makes sense because these are toxins that if they get into the animal's own bloodstream, they could cause a lot of trouble because their targets are generally the kinds of things that are shared among all animals. So whether they're feeding on lizards, birds, or mammals, most of their toxins work pretty well across those various groups. And they, being backbone animals themselves, their venom toxins could be very dangerous. And it's been observed, oh my goodness, probably almost 2 centuries ago that snakes were resistant to their own venom. But it's only in the last few decades that we've started to get a handle on how that is. And one of the ways we can study this, or we can at least demonstrate this, is we can take the serum of a rattlesnake, for example, and mix it with venom and see whether or not, for example, it can protect an animal from that venom. So a simple assay to do that is with mice, shown here schematically, because I'm not going to show you real mice. But it's been long observed that rattlesnake serum inhibits the lethal activity of rattlesnake venom in mice. So if you just give mice venom alone, a lethal dose of venom, the mouse dies. a sublethal dose, you can see lots of morbidity in the mouse. But if that venom is mixed with serum, the venom is inactivated. And we know that one of the really important categories of toxins that's inactivated by serum are metalloproteinases. So this is just a titration done by Fiona Yukin in my lab that shows that as you give just a little bit of mixed venom with a little bit of serum, the metalloproteinase activity of the venom really disappears. It's nearly fully inhibited. And so it's been really interesting to try to identify what in the rattlesnake serum is capable of inhibiting metalloproteinases, as well as things that inhibit other toxins. And we've identified some of them. And again, I'm just going to show you these little graphs that show you the activity of these proteins. I'm going to call one there. We're just going to use the name FETOA for them. And this protein that we've dubbed FETOA3, it's a major inhibitor of venom metalloproteinase activity. It turns out just a relatively modest amount of that protein can almost completely inhibit the measurable metalloproteinase activity of Western diamondback rattlesnake venom in this case. It turns out the rattlesnake makes 5 FETOAs. So one of the other FETOAs, for example, FETOA5 has some activity. A couple others do not. And we're still trying to discover what their activities might be. But if we then again look and we ask the same sort of question about novelty. Okay, the snakes have come up with these toxins. They have these inhibitors. Where do the inhibitors come from? Well, it's a very similar story. Just look at the color coding here. If you look at where FETOA is, you and I all have one copy of the FETOA gene. Sharks have it, platypus have it, mice have it, chickens have it, turtles have it, lizards have it, boas and pythons have it, etc. But the rattlesnake's got 5 copies. So same sort of story. It has taken a protein that's been around for a long time. That protein is involved, for example, in mineral metabolism and in us and in other backboned animals. And it's hijacked it to use it as a self-defense. So the same story here is the venom antitoxins that are in the serum. They're a product of co-option. FETOAs and other antitoxins are derived from ancestral serum proteins that have been co-opted. They've been recruited from other physiological functions to be high affinity toxin inhibitors. Again, we see gene duplication and we see molecular innovation. And this might be leading some of you to wonder, could these natural inhibitors be useful in the treatment of snakebite? So I just want to close on this thought that one of the practical possible outcomes of this research is to learn something about how nature controls essentially venom activities, and we might be able to exploit it. And snakebite, while it's, I would say, pretty well managed here in the United States in terms of lethality, we have a modest number of deaths per year, and some years perhaps with no deaths at all. You look across the globe, this is considered one of the sort of most under treated diseases. Huge number of deaths in Asia, a lot of shortage of antivenoms, a lot of difficulty of access to medical facilities, et cetera. So snakes worldwide are responsible for a lot of human suffering, unfortunately, particularly in agricultural fields and things like that. And so, there's a lot of researchers across the world thinking that this is a medical issue that needs to be addressed, and perhaps some of this newer understanding of what's in venoms and perhaps the identification of some of these things that snakes use themselves to protect themselves could be useful for treating snake bite. And we're working on it. I'm going to save you, that's another talk maybe for another time. But we're certainly interested in seeing how far we can take that. So before I wrap up and take some questions, I just want to acknowledge that the kind of work I'm telling you gets done by a very large team of researchers. I just want to acknowledge people who've been with me at Wisconsin and Maryland and part of HHMI, Noah, Matt, Yori, Fiona, Vicki, Sam, Jane. And we couldn't do this work whatsoever without a nationally supported toxin research center at Texas A&M Kingsville, led by Dr. Elda Sanchez. That's where all to just preempt the question, that's where all the rattlesnakes are. It's a federally supported facility for studying toxic animals, and Elda and her colleagues just do a great job of servicing the entire research community with our needs for venoms and tissues and expertise and things like that. So I thank the brave pioneers in my team, and especially the collaborators. And I thank you for listening, and I'm happy to take some questions.
Thank you so much. That was wonderful. If you can come and unshare, I'll stick a spotlight on you. And then I'll open it up for some questions. We do have a couple of questions. We actually have Joe wanted to know where you went to high school. Let's just throw that out there.
Oh, I should ask Joe. Can I ask Joe why you'd like to know where I went? I'll tell you, but I'm just curious. What makes you curious, Joe? And you can just unmute if you just want to talk to me directly. Is Joe gonna tell me why he wants to know?
Yeah.
See where.
Sure.
I was born and raised in Toledo, Ohio.
Oh, here we go.
So I wanted to know if you were related to John Carroll.
Well, I'm not. I mean, maybe some John Carrolls in the deep past, but come on. I got it. So Toledo, Ohio. So here we go. Yeah, I'm from Toledo. So let's go with this, Joe. Where did you go to school?
St. Francis de Sales High School.
Joe, what year did you graduate?
You're going to ask me that?
Yeah.
1960, what is it? Yeah, 1968.
All right. 1977, Joe, St. Francis.
Oh, a fellow knight.
What is the chat? I know everyone else on this Zoom is probably going, what's going on here? But this is just, I mean, what a remarkable, what a treat, Joe. So did you know I was from Toledo? Is that why, or you knew a Carroll?
You mentioned it. You
Yeah.
mentioned it in the talk.
Oh, I did. I mentioned Toledo. Yes, there we go.
You mentioned Toledo.
Yeah.
And there are very few people that are around Maryland that come from Toledo, Ohio. Most Toledoans stay in Toledo. So it was interesting.
Yeah, I forgot that I had just even mentioned. I was talking a little bit about how I got into snakes, and I got into snakes by catching snakes out on especially the shores of Lake Erie and a few forests around Toledo. But yeah, so everybody, you've now figured out that of all the coincidences in the world, Joe and I graduated from the same relatively modest-sized high school in Toledo, Ohio, 9 years apart. So I was wondering why you were asking, but there you go. So you must have, there were some Carrolls in Toledo. In fact, I knew another Carroll who wasn't in my family, but distantly. And of course, there's a lot of Carrolls all over Maryland. And at some point, a lot of them came from County Offaly in Ireland.
Okay.
In different waves. I'm not in those early prosperous Carrolls that came to Maryland. I'm in the later potato famine Carrolls.
Okay.
The kid that went
Understood.
Yeah.
Understood.
Yeah, great.
Well,
What
they
a
call
treat.
Baltimore. They call Baltimore Smaltimore, and I'm going to continue. That's it. This all happened in this, so it's a small world.
Well, I was prepared for him to know. I forgot that I had mentioned Toledo, but when he mentioned high school, I thought, okay, now we're probably dealing with a Toledoan, but to be the same high school is remarkable. So that's great.
I'm going to get to you in just a second, Jocelyn. I got one question here. Mark wanted to know, what is serum?
Oh yeah, so serum. Take out the blood cells of when your blood gets drawn. We distinguish two things. Okay, so when your blood gets drawn, if you spin out all the cells, we'll call that plasma. If you let it clot, we'll call what's left over serum. So it's really the yellow liquid that's in your bloodstream. I should have made that clear. Sorry about that. But yeah, it's essentially what's carrying all the stuff around your body in your bloodstream that isn't blood cells. So for most of us, about 45% of our blood volume is red blood cells. And a lot of the rest is liquid. And in that liquid, this is how nutrients get around. This is how hormones get around. This is how antibodies get around to fight defense and things like that. They're carried in your serum. And so serum is the main component of your circulatory system. So these antitoxins that the snakes make are circulating out through their body in their serum, and they're sort of there as sponges, just in case some venom leaks, either from the venom gland, or maybe in a tangle with a prey, or maybe in a battle with another snake, or an accidental bite, whatever it might be.
All right, go ahead, Jocelyn, on the screen. You can unmute.
Thank you, ladies and gentlemen. I just have three things. One, is it true that snakes evolved venom? Two, are snakes ancient lizards? And three, I think that's it.
Yeah, so great. Thank you for the questions. That's really cool. Yeah, snakes evolved from lizards. So the ancestors to snakes had 4 legs. And what we think happened was that there was a time or there was an opportunity to live, for example, a bit underground. And if you're going to burrow underground, it turns out having a really a legless body can be an advantage because then your legs aren't getting in the way and you can, slither in and out of tight spaces. And another thing to sort of support that is you may know that there are legless lizards as well. So leglessness has happened several times in lizards. One group gave rise to what became all the snakes. But there are other groups, there are legless lizards, in various places on the planet today. So we've sort of seen evolution repeat itself of lizards evolving, a legless body multiple times in the course of time. And yeah, so snakes evolved venom and what snakes have come up with. So the ability to deliver venom through fangs, snakes invented that relative to other things. And then as I was explaining a bit tonight, they've come up with lots of different kinds of venom. And in fact, they're inventing out there now. I mean, the story is not over, that they're out there, hunting prey and all that sort of stuff. And this evolutionary battle which has been going on for tens of millions of years, still continues today. So I just gave you examples of some of the different varieties of venom out there, venoms that cause the animals to hemorrhage, venoms that cause the blood to clot, venom that causes the nervous system to shut down. So snakes have been incredibly inventive when it comes to venom. But thanks for your question.
So is that how that group of snakes lizards became snakes that had to learn to slither?
Yeah, to slither as burrowing animals and sort of slipping into cracks and things like that. Yeah, they if you think about, for example, if you've ever seen like a video of a snake like burying itself in sand, snakes are incredibly great. And when things are really soft, like leaves and sand and things like that, snakes just slither in and out of that so easily and pretty quietly, right? So there's probably both an advantage in terms of the spaces you can get into, and probably getting into them without being detected. Because, other animals that are in the leaves or other animals that might be in the sand, they're sensing things that are, walking around and tapping the ground. But I think snakes are pretty sneaky in the way they can move around. And that's why we think they evolved a legless body.
Thank you.
Okay,
Most
you.
welcome.
All right, and we have another question. So Timothy asks, since elapid snakes feed on other snakes, are these snakes also resistant to their own venom?
Yeah, it's a great question. Thanks, Timothy. So I think it's a bit understudied. So elapids are a big branch of venomous snakes. They include the cobras, the kraits, all those Australian snakes I was talking about are elapids. It's less studied. I think that things like king cobras, which feed on other venomous snakes like kraits and things like that. They can eat big old kraits, etc. They're resistant to their prey's venom. I assume they're resistant to their own venom. But it's really not as well studied as the serum-based inhibitors are. Some of that resistance in the elapids, the neurotoxic elapids, is that their their nervous system has evolved, and the termini, their nerves, have evolved so the toxin can't bind. So there's one way you could be resistant to your own venom, is that you could sop up the venom with something that's circulating around the blood, like I showed you in the rattlesnakes. The other way you could be resistant to your own venom is that venom doesn't act on your own body very well. And some of that's going on in the neurotoxic elapids, is that they are resistant to their own venom because the neurotoxin does not hit their nerve channels. So, but in terms of what is circulating in the blood of some of those elapids, that's really understudied. So I can't tell you, for example, if taipans are resistant to their own very deadly venom, like the procoagulant venom. I don't know. Not that easy to study. You're like, well, somebody should find out. And I agree. Some of these animals aren't too easy to study. Like for us to get material of the taipan and the brown snake was a really complex process. For Australian researchers, it's a little bit easier, but others, I'm sort of amazed. Maybe I'll put, Timothy, I'll put it this way. I was first interested in some of these questions when I was a kid, which Joe has now revealed was a long time ago. And decades later, so I didn't spend most of my, the work on snakes is really about the last 10 years. I worked on other questions in biology and evolutionary biology for decades. So I came back to snakes as kind of a first love and some questions that I wanted to have answers to that were still open, that were 40 or 50 years after I got into snakes. So it's not a huge field. So there's plenty of room for researchers to ask questions. And you can see that the answer to your question could be very valuable to know, because if we knew how some of these snakes, I mean, like cobras, cause tremendous loss and suffering in the world. If we knew how to defend against them better, that could be really practical knowledge.
Great. Mark asks, what causes molecular innovation?
Okay, so it's the same sort of, it's the general evolutionary process. So what happens is this, and this is, if you haven't been exposed a lot to evolutionary thought, it's gonna kind of twist you 180 a little bit. But we're pretty familiar in sort of like human engineering. We think about like trial and error. You make something, you see how well it works. Evolution kind of works by error and trial. The error is this process of mutation. Every time a new individual is born, there are mutations in that individual that did not exist in either parent. This is true of humans, this is true of snakes, this is true of everything on Earth, every plant, etc. In every individual, every new generation, there are mutations. And those mutations, many of them cause no change in how the animal or plant performs whatsoever. DNA is a big library, and single letter changes across the DNA don't necessarily cause anything. But here and there are mutations that will change the performance of something. And if that performance is advantageous, it turns out that that mutation will persist and it will be passed on through the generations. So molecular innovation happens when mutations that occur just happen to improve the performance of something. Let's say it makes a toxin a little more potent, or it increases the amount of that toxin that's being made. And you can imagine that's how these animals get their meals. And that is right at the sort of crux of life, right? Whether you are efficient at getting your meals in the wild is going to determine how long you survive, how many babies you make, etc. And so venom is under immense pressure. And so the mutations that occur that tinker with the venom, they're being tested sort of out in the wild all the time. And we can see that variation when we look just snake to snake. If we look at 8, 10, 12, Western diamondback rattlesnakes, even caught in the same county, we can see differences in their venom. There's a lot of variation out there, and that variation is created by mutation. And and they're being tested all the time. So I hope that's a decent enough answer, but if you want more, I can refer you to some more reading.
Thank you. Duke has a question about snake bite tourniquets. Do you want to ask the question, or do you want to, is that the question?
Maybe the question is the use of tourniquets, which has been, I think it was standard practice for snake bite for a while and then out of favor.
Go ahead.
My question is, sorry, we were figuring out how to unmute it.
my question was, like, I know for some snake bites, you want to apply a tourniquet to stop the venom from spreading, but some you don't want to apply a tourniquet and let the venom spread so it's not really bad in one spot. Why, like, what's the difference? Can you tell me, like, a little bit of the difference of the, like,
Yeah.
what's the difference?
I think, let me tell you the thinking about it. Okay, so most bites happen on the like the hands and the feet, right? Because you're
Yeah.
either looking or you're reaching the wrong place or something like that. So we call these extremities, right? So they're away from the main body because the stuff you really worry about are like your heart, your brain, your lungs, right? You don't want to get venom to go to those tissues.
Yes.
So the thought was for a while that a tourniquet could keep the venom away. But the trick about that is that some of these venoms are very destructive and you're also cutting off, circulation to that hand or limb or things like that.
Yeah.
So I don't know the current thinking. And like, I can't tell you what the current recommendation is. I wish I could tell you the current recommendation for like treating a rattlesnake bite. I think it's not tourniquet. But I don't know if that goes for cobra or for, something like a taipan bite. The main thing is antivenom and get people to the hospital as fast as possible. So time is of the essence for the most dangerous snakes. Time is of the essence. And as soon as you can get people into supportive care. So if they're, flown, go ahead.
You can ignore them.
Sorry, my little sister is asking me questions.
Oh, okay. So I think that I don't think there's one answer, although my sense is that the medical profession is shying away, is saying no, don't use tourniquets because for people without medical training, it's risky. And the main thing they're saying is get people to medical attention as fast as possible. That's the important thing. And that's why it's like, hiking alone where there's a lot of, venomous snakes, et cetera. Those are risky things because you like to have a buddy around in case something happens. And, funny, I mean, you may know somebody or you may have heard stories. People here on the Zoom may have heard stories, but one of my colleagues was walking on one of the islands in the Potomac down in DC, and he was in sandals and a copperhead got him. And, and he was, he was off to the hospital quickly. So, it does happen. Fortunately, in the United States, antivenom is spread all over the United States. The poison control centers know where it is. The hospitals know where it is. And so if you can just get person to a hospital fast, the outcome's going to be pretty good. We're in pretty good shape here in the United States for care of snakes.
Yeah. I've seen one or two copperheads when I'm looking for water snakes. I stay away. I don't want to get myself into that.
Good for you. Yeah. I mean, it's good to respect them and to know the difference. I certainly, if you like snakes like I do, it's, you probably understand a lot of people, when they see a snake, they think, let's kill it. But of course, that means a lot of harmless snakes get hurt. But I certainly understand people's worry. People with pets, there's a lot of, especially dogs, get bitten by snakes a lot because they're curious. They confront the snake and they get hit. There's veterinary antivenom for pets as well. So I'm glad you like snakes, and I'm glad you're wise enough to stay away from the venomous ones because you don't need that.
I had a quick question.
And your parents don't need that either. What's that?
I had a quick question also on Preston's dad. Was curious how the evolution of the DNA in other poisonous species outside of reptiles it compares. Is it as unique as it is in terms of the single gene having all of those mutations, and do they look the same in other poisonous animals?
That's a great question. I'm trying to think, what's been well studied, and probably one of the best studied groups are these cone snails. Venomous cone snails, which a lot of people like to collect beautiful shells, and it can be pretty dangerous. They're a little bit different story, and yes, you can see the venom toxins in the genome, so you can always see what's there. But some of those toxins are really short little proteins, and they've come from like the pieces of other proteins. It's a little more, that's the best way I can say. I mean, I think some of the concepts of what I'm saying here are sort of generally applied, but in detail, the way I look at the cone snails, they've been doing a little more raw inventing with building blocks of proteins. And yeah, I guess I would just say that there's a little more chemical invention in the cone snails than you would say in the snakes. The snakes are really using things that are very closely related to body proteins, where I know the cone snails have come up with things that don't function in cone snails. They've come up with unique offensive weapons that don't have a counterpart for what works in the body.
So the evolutionary pressures are the same, but the actual evolution of the genes is completely divergent, unrelated.
Yeah.
Okay.
Some toxins are really short protein sequences. So some of the toxins I was telling you about, like the metalloproteinases, they're 400 or 500 amino acids long. They come from proteins that are 400 or 500 amino acids long. But there are some toxins that are just short little 10 or 12 amino acid peptides. It turns out nature can generate those things pretty readily, and there doesn't have to be an existing sort of precursor to it. So there's
it doesn't bear a resemblance to a protein that's naturally occurring in your body. Okay.
Yeah. These so things that are more peptide like venoms, there's a little more invention from scratch.
Okay, thank you very much. We really enjoyed it.
Yes,
Yeah, that's a great question. Thank you.
There's a question for Preston. What kind of snake is that?
This is a ball python.
Hold it so they can see it on the.
Wait, put them on. I gotta change mine so I can see us. Tell us about your snake.
She is she's a ball python, like I said. She is about she is about a year. I got her when she was 3 or 4 months, I'm pretty sure. No, going in my sleeve again.
What a beauty. And
Yeah.
what a great, what a great snake. Was did you keep snakes before the ball python?
No, this is my first snake.
It's a great way to start. It's a really gentle animal, and look at that, beautiful and gentle, and it will only get to be what, maybe 5 feet.
They actually get 7 or 8 feet. Sometimes that's about average full grown size for males, but she is a female, and females typically get bigger than the males. But she and her siblings are underweight for their ages for some reason. It's just all her siblings are underweight for some reason. We're trying to, she's like a little over 200 grams. She's supposed to be 500 grams.
Okay.
So yeah, she's not like she's still full of energy. She's still acting normal. She's just underweight like her siblings for some reason.
Do your friends like her?
Yes.
Yeah.
One time, my little sister does cheerleading, and one time she had a party with like 30 kids. They all held her with no problem. Last night we went to the Herpoween thing, and people held her. She was nice and calm. She was a little tuckered out from car rides. She's not a big fan of car rides. They tire her out. But
somebody asked, what do you feed her?
Oh, we feed her frozen thawed mice right now.
Yeah. Well,
and when she's full grown, we'll be feeding her jumbo mice.
Yeah. Well, you're doing a lot to help people get past their fear of snakes, because you don't see snake. A lot of people don't get a chance to see snakes in the wild, and they're not
Yeah.
quite sure what they're like. So hopefully she's helping people understand what snakes are like, and maybe people can like them a little bit better.
Definitely.
Yeah, good for you. Great.
And
Thank you.
it
And way to go,
Thank you.
Dad.
is illegal in Maryland to kill any snake. It is
Yes.
illegal to kill any snake
Good.
in Maryland. That's the law.
That's good.
But still, they you still have to go to a very remote place to see any because from the past of people killing them,
Yeah.
they haven't recovered yet. And I really enjoy this because I went to Arizona for a week not long ago, so I memorized every single snake there, every single snake and most of the lizards there. So I knew a lot about the rattlesnakes that you're talking about.
Yeah, Arizona is a great place for snakes. It's really,
Yes.
really amazing.
Really remote, so no one finds them and kills them in the first place. Although we went in winter, so we didn't see any, which really disappointed me. But what made up for it? Me and my dad went to a trip to the Dominican Republic, and somebody yelled, "Ah, it's a snake! Kill it!" And my dad called me. He said, "Preston, come over here." And I saw this vine snake climbing on a fence right beside a river. And I got special permission. The guide let me before we left, because we were on a dune buggy trip. Before we left, he let me hike like a quarter mile up where no one would find the snake, hopefully, and I released it. And that was really good. I held it in front of everybody. I showed them this wild snake. I can pick it up and it's not biting me. It's just it's scared right now.
Yeah.
Yeah.
That's good. They need protection. They do important jobs out there, and there are fewer of them than there used to be. So
Yes.
good for you. Appreciate it.
All right. We have one more question, I think, is how do the antivenoms, and I think you sort of went over that, stop such devastating effects?
So the antivenoms that are used today, they're all made in a similar way, which is we take animals like horses, sheep, and we inject them with doses of venom, moderate doses of venom, sometimes inactivated venom, and they make antibodies. And so we harvest those antibodies from the serum of those animals, and we put that into people or dogs or cats that have had snake bite. So antivenoms have been made that way for 125, 130 years. So that's what you'll get if you're bitten by a snake, is the antivenom will be antibodies to venoms that have been made in animals. That's different than the natural proteins that I was talking about, which are the snake's own defenses. Those aren't even antibodies. Those are really tight binding molecules that inactivate the toxin. So different strategies, and there may be some advantages to using these natural occurring proteins in addition to the conventional antivenoms.
All right. Dr. Carroll, with the honey badger, don't they get bit by snakes? Are they just tolerant of it? Is that part of the arms race that we see going on?
Yeah, there's a bunch of animals, mongoose, honey badger, mammals that have taken on hunting these snakes. They're often resistant, and they're resistant in a couple ways. Sometimes it's because they've actually, for example, their nerve channels are actually the venom can't act on their nerve channels as well as they can on other animals. And so they can kind of shake it off. If you've seen some video of that, you can see the honey badger kind of, you know, take a nap, but it survives it. So they're resistant enough that they can hunt the animal, survive a bite, and maybe eat the animal. They're kind of living there on the edge. Opossums are resistant to pit vipers, so opossums can hunt rattlesnakes and things like that here in the U.S. and they're resistant. So yeah, you often see when you see resistance, it's not 100%. It's good enough that they can get by if they happen to get bitten, because being bitten is part of the hazard of hunting these snakes. But often you can see tons of videos with the mongoose where they don't get bitten. They're quick enough to dodge the strike, and then they're fast to jump on the snake and kill the snake. So it's amazing how many animals have found a way to prey on venomous snakes, considering the dangers that are involved there. Also, sometimes in some animals, they have it's the way their body is. So some birds that hunt snakes, they're not actually resistant to the snakes, but they have no circulation in, for example, their lower legs, and nothing for the sort of the fangs to sink their way into. And so, if you see a naked leg on a big secretary bird or whatever, and they're using their feet to stomp the snake or talons to grab the snake, they're actually not resistant to the snake, but they have a hunting method that the snake can't strike them in a vulnerable spot. You can see this in some other animals too. If they're really good at getting to the neck of the snake, they neutralize the snake before the snake can bite. So there's sometimes just physical defense mechanisms rather than a specific resistance.
I love those secretary birds.
Oh, they're elegant. Yeah, they're elegant. Breaks my heart that they feed on snakes, but it is how nature works. Yeah.
Any other questions this evening for Dr. Carroll? If not, thank you so much for spending the evening with us on Must Learn Thursday. We are all grateful for you sharing your knowledge and expertise with us this evening.
My pleasure, everybody. Have a good night.
Good night, everybody. See you next week for Odolist.